Another aspect of plasmid, it is partitioning and segregative stability. So when cell divides, its nucleic acid that included bacterial chromosome or plasmid DNA, they are segregated equally into the daughter cells. But under some circumstances, it cannot happen that during the course of cell division, the plasmids, they are not transferred from the parents to the daughter cells. So if the plasmid it is lost during the cell division, so it is called as segregative instability. That is, plasmid it is not stably maintained in the daughter cells. So most of the plasmids that are natural, they are stably maintained. That is, they are transferred into the daughter cells from the parents after subsequent cell division. But for some plasmids, it is not the case. So I can quote here the example of a natural plasmids like whole E1 that is stably maintained. The reason is that this stability or the segregation of the plasmids from the parents to the daughter cell, it is controlled by the region of the plasmid which is called as partition or the PAR region. So if PAR region it is there, then it can control the transfer or the segregation of the plasmids from parents to the daughter cells during cell division. But it may be not the case if plasmid it is artificially synthesized and especially if lack the PAR region. For example, a plasmid PBr322, it is a derivative of col E1, and if the PAR region it is not introduced, then the PBr322 it will be lost with subsequent cell division. Although PBr322 it is a high copy number plasmid, but subsequent cell division result in its loss because there is no PAR region. But on the other hand, if the PAR region from another natural plasmids like PSC101, it is introduced into the artificially formed or constructed plasmid like PBr322, then it can be stably transferred. Another factor that can mediate the segregation of the plasmids, it is the superhelicity of the plasmid DNA. If it is present in superhelicity form, then it can be stably maintained in the daughter cells. For example, if mutants of PSC101 plasmid they are developed that lack superhelicity as compared to their wild counterpart, then it cannot be stably maintained into the daughter cells. So it indicates that another factor that is important, it is the superhelicity of the plasmid. In addition to that, if we use some DNA gyrase inhibitors, then it can also result in the plasmid instability, and especially if mutations they are introduced in those genes that encode DNA gyrase. So these are different factors like the PAR region, superhelicity of the DNA, and even the mutations in the defective plasmids like PSC101. It can also mediate the instability of the plasmid so that they are not stably maintained in the daughter cells after subsequent cell division.